Thank you for coming. We'll get started. So it's my absolute pleasure today to be introducing you to Dr. Annie Page. Annie is an associate research professor and a clinical veterinarian at Florida Atlantic University down in Fort Pierce, because she's actually stationed at Harbor Branch Oceanographic Institute. And she focuses primarily on large marine mammals, so I think whales and the like, and on sea turtles as well. And she originally obtained her BA up in the College of Charleston. After that, she went on to obtain also a BSc from the University of Georgia. She seemed to quite like Georgia because she stayed there for longer to obtain a veterinary degree, so her DMV also from the University of Georgia. And even after all of that study, she still wasn't finished with academia and decided that she would also really like to have a PhD. So she then spent about 4 more years at the University of Georgia obtaining a PhD. So she's both a qualified veterinarian and a qualified scientist. And she was finally coaxed away from Georgia to join us here in Florida by Florida Atlantic University. So she joined Florida Atlantic University back in 2016 as an assistant professor and as a clinical vet for Harbor Branch. And she has rapidly been moving up the ranks ever since. We don't have time to go into all of the various titles and roles that she actually holds down there, but it's impressive and it's a lot. You'll just have to take my word for it.
It's like 17 hats.
She still doesn't say no to new ones, so. And in addition to all of those roles she has at Harbor Branch, she also has held and continues to hold a number of positions in small animal veterinary clinics, in museums and for zoos as well. And that includes the Puerto Lopez Sea Turtle Rehabilitation Center in Ecuador. So as you can see, she's very far-ranging. Her major research interests include epidemiology, pathogenesis, eco-immunology, ecology of diseases in marine mammals and sea turtles. So it's a bit of a mouthful, but essentially diseases of marine mammals and sea turtles, which she'll tell us a bit about shortly. And she also has a really diverse range of talents. So you can find Annie anywhere from in a clinical vet setting through to a lab bench conducting science, or even out conducting stranding responses and necropsies of marine mammals on beaches. And she's also been a great friend to our sea turtle hospital here at the Whitney Lab since we opened in 2015. I've had the pleasure of knowing her and collaborating with her since we opened our hospital in 2015. And we both conducted research on sea turtle fibropapillomatosis tumors, but Annie actually predates me in this field because that was the main focus of her PhD as well. So she has a lot of broad-ranging experience in a whole range of disease topics. And I'm really delighted that she's been able to take some time out of her very impressively busy schedule to talk to us here today. And she's going to speak to us about sea turtles and marine mammal health assessments and how they provide insights into marine ecosystem health. So welcome, Annie.
Thank you. Thank you, guys. Can you hear me okay? I don't know if this, okay, there we go. Thanks, Dave, so much for that great introduction. You really boosted my ego, made me feel pretty good. So thank you, I appreciate that. And thank you all for coming, for joining us tonight, taking time out of your day to talk about sea turtles and marine mammals and how evaluating their health helps us understand better ecosystem health, especially coastal marine ecosystem health. Okay, so before I get started, I want to acknowledge all of my collaborators and co-authors. All of my work is very collaborative. None of this work could be done by me alone, including I collaborate pretty strongly with Dave here and the folks at the Whitney Lab. Also all over Florida, the Southeast, and kind of the world a little bit. And the reason that we all do this work is because Florida's marine megafauna need our help. If you guys watch the news, you're aware of this, that human activities impact marine animals every single day, especially in heavily populated places like Florida, where humans are constantly interfacing with coastal marine wildlife. So they're subject to all sorts of threats, including entanglements, plastic ingestion, exposure to harmful algal blooms, And then even infectious diseases like fibropapillomatosis, which is one of the things I'm going to talk about today. Okay, so what we do at, I work primarily at FAU Harbor Branch down in Fort Pierce. We're an Oceanographic institute with 32 principal investigators. Each of us runs our own program there. And I run the Marine Wildlife Veterinary Medicine and Research Program. You'll notice most of the stuff I do is like a mouthful. I always overdo everything. I don't know why I do that. I'm just an overachiever, but anyway, it's a mouthful. What we do is we provide animal rescue services. We cover about 75 miles of coastline for marine mammal strandings in Southeast Florida. We provide stranding response. We provide euthanasia when needed for stranded marine mammals. We also provide transport for live and dead marine mammals. We also participate in disentanglement efforts for things like entangled dolphins, and these are multi-agency efforts, and I'm going to talk about, give you an example in a little bit of one that we did that was really cool. Every animal in our stranding area that dies, we conduct a necropsy on. A necropsy is like an autopsy for animals. And we try to understand things like why they died, things that affected their health during their life, try to understand more of their life history, things like that. I also hold a permit to conduct sea turtle stranding response in my area. That's just something I do for fun. And again, providing transport and stranding response for sea turtles near where I live. Okay. My research focus, Dave kind of touched on this, but the focus of my research program is basically understanding emerging infectious diseases of marine wildlife, how humans impact marine wildlife health, and then looking at ecoimmunology, so looking at how the ecosystem health affects and relates to animal immunology. The immune system is my favorite organ system. Blood is my favorite organ. You'll probably notice that through the talk. So I always get into these ecoimmunology topics. All of the work that I do is done within a conservation context. So essentially the goal of our work is to provide more information so that we can protect and sustain wild animal populations for the future. And also with the understanding that these animals, at least the animals that I work with, are considered sentinels of ecosystem health, meaning that the health of these animals is dependent upon and reflects the health of the ecosystems that they inhabit. And just going into some of these words for you, definition of disease ecology. This is looking at host pathogen interactions within the context of their environment and evolution, and looking at disease dynamics basically in time and space, and understanding the dynamics kind of in a more of a three-dimensional sense of how diseases affect populations on the population scale. I also am interested in pathophysiology, so like how do diseases make animals sick? What are the things that happen inside an animal's body when they start to get sick, when they start to recover? That's something that just interests me. And then ecotoxicology, I'm not a toxicologist. I wasn't doing toxicology until I moved to Florida. But because Florida, now I do a lot of toxicology. Again, because there's so much coastal area here in Florida, and there's all these pollutants that are constantly mixing in the coastal areas. You're going to notice again that a lot of the work that I do is based in toxicology topics. And then essentially, I'm going to be talking to you guys about health assessments of marine wildlife. And this is really with the understanding, that wild populations cannot be considered healthy unless the animals that make up those populations are themselves healthy. And so I'm a veterinarian, but I work with a lot of wildlife biologists. Biologists like to say, biology, we look at the population, veterinarians look at the individuals. But if you're looking at a population, the population consists of individuals, and especially if it's a threatened or vulnerable species with lower animal numbers, the health of those individuals actually does matter. And that's kind of where I come in. So when we do health assessments, we're looking at the health of essentially a lot of times healthy animals. And the reason that that's important is because we're conducting surveillance, we're creating a baseline of the health of a population so that later in the future, God forbid something happens, a disease outbreak, an oil spill, a harmful algal bloom, things like that. And then we have a baseline to compare against for future events. And this allows us to manage their populations more effectively. And so in this talk, I'm going to give you some examples of how I conduct marine wildlife health assessments in my program. There's lots of different ways to go at it. The things that I do, I work with people who rehabilitate wildlife, such as the wonderful folks here at the Whitney Lab. I also, as I mentioned, do stranding, so we do health evaluations of all of our stranded wildlife as well. And then the hardest, most logistically challenging, and the most fun is evaluating the health of free-ranging wildlife. Okay, so the first one is looking at some examples of projects that we've done with rehabilitating wildlife. So a lot of people will argue that the minute a wild animal comes into a captive care setting, they're no longer considered wild. But that's not necessarily true, especially if the goal is to release them again back to their environment. And, you know, so they are really kind of still wild animals. They are wild animals. And when we have them in this rehab setting, it's usually for a temporary period of time. And it's really a great opportunity for us to have access to things about them, to understanding things about them, including their physiology, proper nutrition for them, understanding normal and abnormal behaviors, evaluating and diagnosing and treating different diseases, toxicology topics, and then improving our medicine of these animals, such as better surgical approaches, better antibiotics, all of that stuff. And so rehabilitating animals, it's not, you know, they're not experimental animals, but we still can gather tons of information taking advantage of this temporary access to them. And then let's see, access to wildlife provides valuable information. Again, this is kind of what I just said, but standardizing different medical approaches, looking at evaluating disease prevalence within our rehab population, and all of that kind of gets distilled down and allows us to make these population-wide inferences that can then be used to better rehabilitate wildlife in the future and also better manage wild populations as well. Okay, so an example of how we've done that in some of my work and some of our work actually with Dave as well here at the Whitney Lab is with this terrible disease called fibropapillomatosis. Can you guys raise your hand if you are familiar with this disease? Okay, so about half, that's good. So yeah, this is a debilitating, transmissible tumor disease that mainly affects green sea turtles. It has reached epizootic status, meaning like epidemic status in Florida and in several places around the world. And it's actually considered a panzootic, meaning, or like a pandemic for animals worldwide. The disease really consists of these cutaneous or skin and also internal tumors. The tumors are histologically benign, so technically they're considered benign, but they can behave really more or less malignantly. They can be aggressive, infiltrative, multifocal. Turtles will have tumors in lots of different places all over their body at one time. Turtles with severe FP can become debilitated, emaciated. They become colonized by excessive epibiota. They can have secondary infections like bacterial and fungal infections, and that all is bad for the animal. So FP, which is what I'm going to call this disease, is associated with a virus called Chelonid alpha herpes virus 5. And this virus has been the focus of my work for over 10 years. The strongest association, people have tried to associate FP with different environmental factors, and that's actually been the focus of some of my more recent work. But the strongest association so far that we know is these near-shore shallow water embayments is where the turtles tend to have the most FP. In Florida, FP does appear to be increasing, although it's difficult to tease apart the booming green turtle population in Florida. The green turtles are doing really, really well in Florida. And along with those increasing green turtle numbers, we've got increasing prevalence, increasing percentage, and numbers of turtles with FP. But overall, some of my work has shown that it is generally slowly increasing in Florida, which is crazy. Okay, so in rehabilitating turtles, FP is bad, and that's mainly because it's contagious. So we know that turtles that have tumors are contagious to other turtles through some previous work, not mine. And so having these turtles in a captive setting requires us to have special quarantine conditions that can be challenging especially for facilities that are working on like a shoestring budget. You need separate facilities, separate materials. Sometimes you have to separate staff hours based on the turtles with tumors, keeping them separated. Also, having FP as a rehabilitating turtle is bad. It tends to, a lot of times turtles will strand for other reasons, but then they also have FP tumors. And this tends to influence their clinical progression in a negative way. It can extend their rehabilitation duration, so making them stay longer, and it just complicates their prognosis. This is an example here. This is a turtle that was rehabilitated for almost a whole year at the Georgia-Sea Turtle Center, and this was her pre-release exam. And the staff noticed this bulge in her plastron, and they CT'd this turtle, and it turned out she had a huge tumor, a big internal tumor, and she had to be euthanized. So that's over a year of investment, resources, time, energy in getting this turtle ready to release. So that's just an example of what FP can do. So one of my first studies on this disease this is actually my first paper a long time ago, 2014, but just did a quick retrospective case series analysis of turtles in rehab with FP at the Georgia-Sea Turtle Center. We actually found some pretty interesting trends indicating that the best thing, if turtles are going to develop tumors in rehab, it will be around 90 days after entering rehab. So we came up with the recommendation of trying to release the turtles within 90 days if possible, and also releasing the turtles in the location that they were, found is a good idea because we kind of want to prevent spreading the disease as much as possible, even though it's pretty much everywhere in Florida. Right now, laser surgery is a treatment of choice for tumor removal, so just removing the tumors and letting the turtle heal on its own. This is Pei. This is a turtle that was rehabilitated at the Turtle Hospital down in Marathon. This is one of my favorite examples. You see this turtle, this is severe FP. The tumors are occluding both of her eyes. She can't see. She has tumors all over her body. But she had some special attention. This is a human eye surgeon who donates her time to help these animals, like in removing corneal tumors. And this is Pei right before she was released. So it is possible to rehabilitate these animals, even with, in some cases, that have severe FP. So it's not always a death sentence to have that. However, I will say that after that initial study, in 2019, we did a much bigger study. I reached out to a lot of my colleagues, and we ended up with over 750 cases of turtles with FP in 4 rehab facilities in the Southeast over a 10-year period. So we did a much bigger epidemiological study of turtles with FP in rehab. And we found that almost half ended up having their tumors removed surgically, and of those, half of those had tumor regrowths within 45 days of the tumors being removed. So the tumors can regrow, and sometimes they regrow more aggressively than they started. We also found that turtles with FP, 75% of them did not survive rehab, but the survivorship, I'll show you that in a sec. The survivorship declined significantly with disease severity. So turtles with mild to moderate FP were, more than 50% likely to survive rehab or be released, whereas turtles with severe FP, more than 80% of them ended up being euthanized. And so this was a depressing study. It's not what I was hoping to find, but it did allow us to make more recommendations about triage as far as like if you're intaking severely diseased turtles, you may not want to use your resources to try to rehabilitate them because over 80% of them end up being euthanized anyway. So again, just some kind of general guidelines that resulted from that work. This is just an example that I put in here of tumor regrowth. So this is a turtle. These are just mild little tumors that were surgically removed. This is immediately after they were removed. And this just shows you 15 days later, the tumors have grown back and they're actually worse than they were originally. So that's part of the disease progress of this disease because it has an infectious component, like a viral component. So you're removing the tumors, but the virus is still there. It's a herpes virus. You guys probably know herpes is for life. You can't get rid of it, and it's the same for turtles. So, you know, even though we remove the tumors, if the virus is still there and the turtle is immunosuppressed, we do see regrowth, unfortunately. So it kind of expanded that study. So that was in green turtles. All of that's in green turtles. They're the species that get FP the worst. But other sea turtle species can also get FP tumors. And so we did a quick, another case series analysis. I reached out again to everyone that I knew, and I came up with 7 cases of FP in rehabilitating loggerhead sea turtles. We found that FP in these animals is usually mild, usually self-limiting, usually does not cause a big problem. If the tumors are removed, they usually stay removed, don't regrow. There was one case that was an exception. This was a turtle named Dina, and she was covered in FP tumors. A lot of this stuff, it doesn't look like tumors, but that's what it was. Also, all through her internal organs. An interesting part of that case was that she had a unique variant, variant D, which is unique to loggerheads of the herpes virus, whereas these others had the green turtle variants. And so maybe there's something genetically with the loggerhead specific variant that causes severe disease. That was just a really interesting case to me, but it was only one, so we couldn't really do a whole lot more with it. We sequenced it, Dave sequenced it for me. Part of my work on FP, this is part of my PhD, I developed a PCR assay, a polymerase chain reaction assay to detect CHHV5. And then I proceeded to use that assay to evaluate all different kinds of samples that we collected from green turtles to try to understand how the virus is distributed in the bodies of turtles with FP. And so we ended up looking at all different types of samples, including blood, serum, skin, scutes, internal organs, urine, feces. I'm trying to think if there was anything else. And then we also looked for CHHV5 in green turtles that did and did not have FP tumors. And one thing that we found that was interesting is that almost half of the green turtles that we tested with that did not have FP tumors did test positive for the virus, showing that there's a lot of turtles around here are carrying the virus, but not all of, and have been exposed to the virus, but not all of them develop tumors. And that's one of the biggest questions I think of FP still remaining is why is that? So I skipped over a bunch of stuff there, but I don't want to get in the weeds. I feel like I'm taking too long. So anyway, I ended up also applying that PCR assay, which detects DNA, viral DNA, to a bunch of samples that were sent to me from green turtles and loggerhead turtles in North Carolina. This is significant because FP tends to be more of a circumtropical disease, so like in much warmer waters, we tend to see much fewer cases of FP the further away from the equator we get. And essentially the further away from Florida that we get. FP was first described in Florida in the 1930s, and it really is kind of like the nexus of the world for FP. I'm going to show you more of that in a minute. But anyway, so we ran all these samples for turtles in North Carolina, and only 10% of them were positive for the viral DNA. But almost 60% of them were positive for antibodies to the virus, meaning that they had been exposed at some point in their life, even though none of them had tumors. So again, showing that the virus is circulating in these populations, but the turtles do not always develop tumors. So there's probably another factor, like a cofactor, that's causing infected turtles to develop tumors. And then finally, what happens when you publish these papers is that people reach out to you and they say, oh, we have a turtle with tumors here and here and here. And I have trouble saying no. So anyway, I ended up doing another study of another sea turtle species, Kemp's Ridley sea turtles. This is the most endangered sea turtle. They do occur here in Florida in lower numbers. They also occur more west, and they nest on the coast of Texas and in Mexico. And the people who work the beaches down there have noticed there have been some nesting female Kemp's Ridley turtles that come on shore and they're nesting and they have tumors on their skin. And that's kind of a new observation. And so now, we did a study where we looked at the viral metagenomics, so really looking at the genetics of the virus in these animals and then comparing it to what we know from other species and other locations. And this was, again, a collaboration with Dave and here at the Whitney Lab. So again, reached out to everyone I know and identified 22 cases of Kemp's Ridley turtles with FP during, a 14-year period. One interesting thing from that study is that nearly half of those were reported in the most recent years of the study. So again, that fits with what people were telling me anecdotally, is that, this is a new thing, we're seeing it more. And a really cool thing that came from that study is that one of the turtles stranded all the way up here in Massachusetts with tumors. And so that turtle ended up being the northernmost turtle that had been reported with FP anywhere in the world. So that was a really unique thing. And again, I think our work shows that it's spreading. So a lot of turtles in Florida, but then also a lot of turtles nesting females. And that's also significant because, as I mentioned, Kemp's Ridleys are critically endangered. So nesting females are super duper important for their population. And the last thing we want is for them to have a deadly tumor disease. I already said that. Okay, so that's everything I have on FP in rehab. So that was just kind of like a smorgasbord of a lot of the collaborations that I've done to try to better understand this disease by looking at rehabilitating animals. So now I'm going to switch gears and give you some examples of some of our work of health assessments in stranded marine wildlife. The first one, this is probably the biggest study that I've done so far. What we did, so at Harbor Branch, as I mentioned, we do all of these marine mammal strandings, we do necropsies of stranded marine mammals, and then we have a tissue bank, tissue archives, going back about 25 years. So I run 4-80 freezers full of cetacean parts. And for this study, what we did is we dove into our tissue bank and analyzed a lot of things for contaminants. So let's see, I already said those things. Cetaceans, cetaceans meaning whales and dolphins, tend to bioaccumulate contaminants, especially lipophilic contaminants, in their fatty tissues. They have blubber, you're probably familiar with this. So they tend to store a lot of various contaminants in their blubber layer and in their liver, these fatty tissues where these things accumulate, and so then we can measure them. So for this study, I again reached out to collaborators. I worked with collaborators in Georgia, North Carolina, and Florida. And we came up with almost 90 cases of stranded cetaceans to work with. Most of them, as you can see, were bottlenose dolphins, followed by pygmy sperm whales. And this represents this really is a good example of what our stranding cases look like. So it's mostly dolphins, some Kogia, and then like a little smattering of all these other offshore, benthic, and weird species. Those are my favorites, is the weird ones. So we did, I had a grad student, Catherine Lowe. This was her master's project. It was a really big master's project. It probably should have been a PhD, to be honest. But anyway, she did a retrospective analysis. We looked at all their stranding data, their necropsy reports, their histopathology reports, and then we ran toxicology on banked liver and blubber samples from these animals. And so the things that we looked for in their tissues included non-essential elements. So most people call these heavy metal toxins. So these are essential, or they're basically elements that have no known biological role. And the ones we analyzed in this study were arsenic, cadmium, lead, mercury, and thallium. So they're essentially pure toxicants. And the reason that we wanted to look at these toxicants is because they tend to both bioaccumulate and biomagnify within marine mammals. And they can cause all sorts of health effects, direct health effects, including organ dysfunction, decreased reproductive success, and then direct mortality from excessive toxicity, although that's probably very rare. And also, they have indirect health effects. They can cause animals to become immunosuppressed over time. They can cause animals to change their behavior, their social behaviors, and it can also change their prey availability, which means that animals have to migrate further, longer, go bigger distances to find their food, which changes all sorts of things from reproduction to fitness to behavior. So bad. In that study, we found lots of things. I encourage you to read it. Anyway, I just have some of the highlights here. A lot of things we found were in bottlenose dolphins because we had the big sample size, so we could compare stuff. We found significantly higher lead, mercury, selenium, and thallium concentrations in dolphins versus pygmy sperm whales. And that difference is probably reflective of a difference in their diets. Dolphins eat a lot of teleos fish, bony fish, just like we do. We share a lot of the same prey species with bottlenose dolphins. And pygmy sperm whales are squid eaters primarily. So we also compared males and females. We found higher concentrations of arsenic in females versus males. We also compared adults versus juveniles. We found higher concentrations of lead, mercury, and selenium in adults, again reflecting their bioaccumulation, biomagnification. And then we compared sites, and we found that dolphins that stranded in Florida were more intoxicated than dolphins that stranded in North Carolina. And this was actually one of my favorite parts of the study. We had four mothers that stranded that were pregnant. And whenever a female strands that's pregnant, we do 2 necropsies. We do a full necropsy on the mother, and then we do a complete necropsy on the fetus as well. So we try to make the most of a really sad event. And then for this study, we compared the mothers to their fetuses, and we found the mothers had more arsenic, mercury, cadmium, and selenium. But then the fetuses, I didn't write it down, but the fetuses did have higher concentrations, for example, of triclosan. Like, this is a big study. I didn't tell you guys everything, but this is an organic contaminant that we looked at. The fetuses had higher concentrations of zinc and copper. And so it just kind of gives us more information about how essential elements, toxicants, and contaminants are distributed in mothers versus their fetuses, and then how that changes over time. So to me, that was really interesting. Okay, so that was that study, and then I wanted to show you kind of a different take on how we use these stranded marine mammals to understand their health. So this was a case study. The first thing was a big study of lots of cases. This one was a single case that ended up being very interesting and sad. But I will say one of my favorite things about my job at Harbor Branch is that in most cases, if you're a wildlife pathologist, when you get a dead animal on your table, what you see is what you get. The information is contained on your table. There's no history. It may be like found by someone somewhere. That, a lot of times that you get very, very minimal history. But for because at Harbor Branch, we also have a really active dolphin photo ID program. So over 600 dolphins in our photo ID catalog. These animals are known. These are dolphins that live in the Indian River Lagoon. They're known. They have, we know a lot about their life histories, their genealogy, their social groups, where they like to hang out, what they do in those places. We have all this information in our database about these animals when they're alive. And so then when some of those animals strand, we have like a complete history. The animal's got a name. We know how many calves she'd had. You know, we know how old it is, things like that. So it's a really, really cool opportunity to have history on a wild animal. At least for me, it's cool. But I mentioned that at Harbor Branch, we participate in dolphin disentanglements. Usually, I don't know if you guys have heard about this, it's often in the news, but it's almost always a juvenile male dolphin that gets entangled. You can infer what you will on that, but they're the troublemakers. They don't mind the rules and they like to cut corners, I think. They're very curious. And so it's usually juvenile males, they get entangled, maybe they have bad habits, like they fish off piers, or they look for fishermen. This was one of those. This animal was tracked, captured, and disentangled in 2015. So you can see he had a pretty good wrap of monofilament around his rostrum. He was captured, it was really pretty deep, and disentangled and successfully released in 2015. After that, the animal was tracked over more than 2 years. So all along the Indian River Lagoon, he was sighted over a couple years, first sighted with his mom and then sighted on his own. So it was like, okay, well, he's doing good. This was a success. Then in 2017, he stranded, dead. And this is what I got on my table. You can see lots of different things. You can see the old wrap scars on his rostrum. You can see that he's very thin. We call this post-nuchal depression or peanut head, so this tells you that the dolphin is thin. You can also see his vertebrae here on the sides. Again, that tells you he's very thin. And then this was all probably post-mortem scavenging from sharks. So sorry for the gruesome images. I should have said something. Warning, there's some gruesome images. This was what he looked like. So this is 2.5 years after the disentanglement. You can see he is disfigured still by the disentanglement. It goes all the way down. His teeth are gone. It's eroded the bone of his rostrum here. And then actually had an amazing technician, Wendy Marks, who went all the way down to the bone, and just to demonstrate the bony erosion and the disfigurement that he had, even though it was considered a successful disentanglement. I'm not knocking disentanglement, I'm just saying he was disfigured. But this is what ended up killing him. Inside their blowhole, dolphins have basically their larynx goes up into their blowhole, and that little aperture, we call it a goose beak. So that's what you're seeing here. This is his goose beak that I pulled from his blowhole. And you can see he has a hook embedded in his goose beak. And here you can see the hook has tracked. So it's been there a long time, and it has tracked through his tissues and caused a lot of damage. And also probably directly relates to how thin he was. Oh, sorry. And so the take-home message from this case, and one of the reasons that we wanted to publish it, it was cool that we had his whole life history, we had the disentanglement history, but what killed this dolphin was bad habits. So he already had the bad habit of being around fishermen when he was entangled the first time. He clearly didn't learn anything from that experience and went back and was probably fishing off piers. If you guys are fishermen, you're familiar with what they do. They wait and they raid people's hooks. And so that's probably what he was doing. And it ultimately killed him. And so for us, one of the biggest take-homes from this case was to just remind people that when you see wildlife, when you see sea turtles and dolphins, when you're fishing, if they're attempting to steal your bait, reel in your line and just go fish somewhere else while they're there. Because even if you're not feeding them directly, you don't want to encourage their bad behavior, because even though you might think it's cute, it could end up in their demise. So, okay. All right, so that was stranding. And now I'm gonna give you guys, this is the last part, just talking about how we assess health in free-ranging wildlife. So this is some of the more fun work, not dead stuff. So I'm kind of a fan of green turtles because of all of my FP work. And so I also work a lot with Loggerhead Marine Life Center down in Juno Beach. One of my close friends is the research director there, so we collaborate a lot. And he's working with nesting turtles there all the time, and so I said, hey, let's do a health assessment of these nesting turtles and develop some reference intervals for them. So that's what we did. We took blood samples and evaluated body condition of 60 nesting female green sea turtles on Juno Beach. We ran the PCR assay for CHHV5. We evaluated them for FP tumors. And we also did serology to look for antibodies to CHHV5. So that tells us if they've been exposed in the past. And what we found was that only 8% tested positive for viral DNA, indicating like an active infection. Almost 30% were positive for antibodies to CHHV5. CHHV6 is another herpes virus in sea turtles. We just looked at that for fun. It's not really an issue. So after that, we waited for those nests to hatch, and when the hatchlings came out, we collected blood samples from them, from those females' nests, and we measured the hatchlings, took teeny tiny itty bitty little blood samples, and we again PCR'd those blood samples for CHHV5 to look for the possibility of vertical transmission. So most herpes viruses are transmitted horizontally, meaning from me to you. But some herpes viruses, like Ostriid in oysters, oyster herpes virus is transmitted vertically, so in the gamete, so from mother to offspring. And so it is possible for reptiles to vertically transmit herpes virus, at least theoretically. So we wanted to see if that was happening. And what we found was that all of them tested negative for CHHV5 in their blood samples. So that's not conclusive that it's not being vertically transmitted, but it is indicative. And I wanted to mention none of the mothers had FP tumors either. And that's typical for green turtles. It's usually a disease of juveniles, not adults. Okay, from that same study, we just, a lot of my work, you'll see I just pile stuff on because it's like, well, when are you ever gonna get this opportunity again? So from that same study, we put data loggers in those nests and tracked the temperature of those nests. And we found that green turtles that came from warmer nests had a higher body mass. They were bigger, right? They were bigger turtles, higher body condition. And they were also, a little bit dehydrated, and they also had a little bit more inflammation than turtles that came from cooler nests. So not that big of a deal, but did allow us to get a few additional data points and results from the same study. And that was another paper, which is good. Okay, so that was to kind of set the stage, to kind of show you guys, you know, that approach of like looking at the mothers and then looking at their offspring, and we can ask a lot of questions about their health by doing that. And so in this study, we wanted to look at leatherback sea turtles and their contaminants. So remember when I mentioned a lot of contaminants are lipophilic and settle into the blubber layer in marine mammals? Leatherbacks are unique sea turtles. They are kind of like marine mammals in a turtle body. They don't have proper blubber. It's not true blubber, but it looks, acts, and functions like blubber. And so we thought maybe these guys are storing contaminants in their fat the way that marine mammals do. So from nesting female leatherbacks in Juno Beach, we took, again, blood samples. We looked at all sorts of stuff in their blood, including we measured for endocrine disrupting contaminants, which are usually lipophilic contaminants from human-made materials. We looked at their proteins, we looked at hormones, and we looked at markers of oxidative stress. Then when the nests of those females hatched, we collected the hatchlings, we took morphometrics on them and took little blood samples from them as well, little teeny tiny blood samples. So you can see we didn't hurt them. And we actually published on the technique as well, so people would not think that we were spearing them. It feels like you're spearing them, but they're okay. Okay. Then after the nests emerged, a lot of times there will be unhatched eggs or even hatched eggs down, or undeveloped eggs down in the nest. And those nests are excavated by the staff at Loggerhead Marine Life Center. That's part of what they do. And so we took some of those unhatched eggs, and we also measured for the contaminants in the egg yolks as well. And then we compared the concentrations that we found in plasma versus eggs. These are the contaminants that we looked for. We kind of wanted to choose like a representative smattering of contaminants that they could be exposed to, including atrazine, which is a very common pesticide. BPA, you're probably familiar with. It's a plastic component. Diethyl phthalates, these are plasticizers or plastic components that are used in a lot of like food preparation products, for example, containers. Nonylphenol ethoxylates, again, plastic components, plasticizers used in things like paint, other plastic materials, and then triclosan, which is used in a lot of human personal care products. So we chose these. These are all different contaminants, and we thought would be kind of like a big representation of what they might be getting exposed to. And one of the things about that study is we ended up with a very low sample size, only 17 females and 20 nests. So that was kind of a bummer. We chose a low year to do the study. But we did get some cool results. We did find that atrazine, BPA, diethyl phthalates, and nonylphenol ethoxylates were detectable in blood samples from nesting females. That might not surprise you, but it surprised us because leatherbacks are supposed to be pelagic turtles. They're supposed to be animals that spend the majority of their life out in the middle of the ocean. You wouldn't necessarily expect them to have these long-term exposures, or relatively long-term exposures to coastal contaminants. So that was, depressing. We also found some of the same plastic contaminants in their eggs. And we found a correlation between diethyl phthalate concentrations in the eggs and hatchling body condition, suggesting perhaps exposure to these plastic contaminants are reducing the growth rates of developing hatchlings in the beach. Okay, so the same, and just want to give you an example, again, the same study we asked a different question of the same data, and we looked at nest temperatures again and hatchling size, and this is the study where we published our blood collection technique for everyone to copy. And we essentially found that nest temperature is related to hatchling hydration status, which is to be expected. Okay, so that was that. That was just some examples of health assessments in nesting turtles and their offspring. And then I have 10 minutes, so I'm going to tell you guys, this is the study that we just concluded. This is probably the best part of the presentation, in my opinion. And I'm working on this paper right now, so it's fresh in my head. But we just finished a 5-year study looking at harmful algal bloom associated biotoxins in free-ranging sea turtles in Florida. So you guys are all familiar with harmful algal blooms. The organisms that create these algal blooms a lot of times also extrude toxins that cause different effects depending on the type of toxin. We call those biotoxins. So especially juvenile green sea turtles are very coastal in Florida. They display high site fidelity in Florida. They stay in one area for maybe even up to 20 years that they use as their developmental and foraging habitats. So in those habitats, they are regularly exposed to harmful algal blooms, probably way more often than we think. And these harmful algal blooms occur, you guys probably know, but in large part they occur because of coastal nutrient runoff and eutrophication of coastal environments. So this study was part of a much bigger study that we did at Harbor Branch. I just did like the turtle component, but I collaborated with, there were 17 PIs on this project, where we looked at algal toxins in all different levels, trophic levels, to compare. And so I don't have the results of the big study for you today, but just there will be a really big paper coming out in the next couple of years where we're looking at everything from the sediment all the way up to dolphins, for example. So this was my part of the study. There were 5 sampling sites across Florida, and we tried to choose like known developmental habitats for green sea turtles. And we worked in large part with a group called In-Water Research Group. So they're already going to a lot of these places and catching turtles. So it was a collaboration. And the sites were Jennings Cove near Fort Pierce, near where I work, and that's on the Indian River Lagoon. The power plant intake canal on Hutchinson Island, which those turtles, they get sucked into the warm water pipes of the power plant, and then they end up in this canal and they're removed. And in the process of removing them and transporting them, we would just take a blood sample and put them in the study. The Big Bend Seagrasses Preserve up in the Big Bend of Florida is, if you have not been there, please go there. It's one of the most beautiful places I've ever been. And then the Lower Florida Keys near Bahia Honda, we've been catching turtles there for 4 years. And then Lake Worth Lagoon, we caught turtles there one year. And that's kind of near West Palm Beach, and those turtles represented our more like our city turtles. Our urban turtles. So this shows you just kind of an overview of catching them. We have a tangle net, or we would do turtle rodeo, depending on, meaning jump on them, depending on where we were. We land the turtle on the boat, get it out. The nets are monitored continuously, so whenever a turtle hits the net, we just go over and get it out. It doesn't stay there. Then we take all sorts of measurements, lots of different measurements. This is a good opportunity for students to get involved. We weigh the turtle, we take a blood sample. We took a scoot sample, so that's keratin on their shell. For a different study, we're using ultrasound to look at their fat depth layer to try to understand body condition. We asked the turtle what it had for lunch. We tag them and mark them. This is temporary marking just so we don't recatch the same animal. Pose with them, and the best part is releasing them. And so we did that over about 5 years, and we ended up with lots of turtles. One of the best parts of that study is we ended up doing a collaboration with a group called Force Blue, which is military veterans, usually like Marines or Navy SEALs, people who worked in the water. And they funded part of our work, and they ended up coming out with us and becoming like amazing turtle catchers as part of our project. So that was really cool, a really kind of unique opportunity for us researchers to work alongside these kind of war heroes, basically. And this kind of gives you an overview of what we found. Okay, let me back that up. So for all of these turtles, we took blood samples, and we tested the blood samples for biotoxins. And we used mass spectrometry. We tested them for a suite of 14 different HAB-associated biotoxins. And we chose the biotoxins based on what we would expect to find in coastal Florida waters after what other people have found. And let me mention, these turtles were considered healthy. They were not sick. They were not showing any signs of intoxication. But we did find that almost 50%, 42% of them were positive for at least one HAB-associated biotoxin. 10% were positive for more than one type of biotoxin, and samples from all 5 sites tested positive for various biotoxins. The most common one we found was domoic acid, followed by microcystins, Lingbiatoxin A, okadaic acid, nodularin, and brevetoxin. So these are all toxins that can also affect humans. So, you know, that's also one of the important components of the study. We did not detect anatoxin A, saxitoxin, neosaxitoxin, or cylindrospermopsin in any of the turtles. And this shows you the distribution of the biotoxins that we did find based on the different sites. And this was really cool to me because, you know, they're kind of different, right? Like this one looks a lot like this one, but not like these two. And so we can start to make inferences about exposure risk in the different sites based on these results. We also tested so that was their blood samples. When we asked the turtles what they had for lunch, we also tested, we call those lavage samples or esophageal lavage samples, and we tested those for biotoxins as well. 16% were positive for at least one biotoxin. The concentrations were consistently higher in the stomach content samples compared to the blood samples, suggesting that they're being exposed through ingestion of the toxins. So that's what we would expect, and it kind of confirms that, which was cool. From those same lavage samples, we sent them out to Dr. Karen Holloway Atkins, who is amazing, and she can tell you what a turtle's been eating based on little teeny tiny scraps of material. And so she looks at this stuff under the microscope and says, oh, this is what they've been eating. And so we were able to like compare the different diets for the different sites and with different levels of biotoxin exposure. I'm not totally finished with the analysis, but we did find, interestingly, that the turtles in the Florida Keys, they're eating what they're supposed to be eating. They're eating seagrass primarily, which is great. 92% of them are eating mostly seagrass. Versus, this is the turtles from the Indian River Lagoon. They were eating almost exclusively red algae, probably because there was no seagrass to eat. And a lot of the red algae species that they were eating, they're listed down here, but a lot of them, more than half of the red algae species that they were eating are considered invasive. So again, this is not great, and we can link it to different levels of biotoxin exposure and various measures of health. So also we found that overall across all the sites we had 151 turtles total over 5 years. 53% of them had FP tumors. And this is probably the most exciting thing of the whole study for me, is that we found a significant correlation between turtles with FP that tested positive for Lingbiatoxin A, which is a biotoxin extruded by Lingbia, which is all over the coastal Florida areas, including all over the bottom of the Indian River Lagoon. And there have been a couple studies in Hawaii from the earlier 2000s where they linked FP in Hawaiian green turtles to Lingbiatoxin exposure. So that's 2 different locations in the world that have made that association. And so I really think there is something to that. And it may be that this is the missing link, the missing cofactor of why not all virus-infected turtles get FP. What is the environmental cofactor? Maybe it's Lingbiatoxin. And so now I have a PhD student who's also a vet, and this is her work. She's going to be following up doing laboratory exposures of FP tumor cells to Lingbiatoxin, turtle skin cells to Lingbiatoxin and see what happens. Okay, real quick, I know I have like 30 seconds, but just to also illustrate how we try to maximize these turtle encounters or the animal encounters as much as possible, get the most information that we can. When we're catching these turtles and taking their blood samples, we were also looking at the immune function in some of them and looking at how immunity factors into FP tumor disease. And we found that turtles with severe FP had significantly decreased lymphocyte activity, so their immune cells are not proliferating in the way that they should. And we also found that turtles with FP had lower natural killer cell activity than turtles without FP. And these are both really significant because T cells, which is a form of lymphocyte, and natural killer cells, we know are what reptiles use for virus immunity and also immunity against tumors. And so this is also really significant, and we're doing a follow-up study now looking at immune function in a much bigger sample size of turtles with and without FP. Okay, so that's all I've got. I'm supposed to do this plug for my organization. Go to our website, check us out. I'm just like a tiny cog in the wheel at Harbor Branch, and there's tons of great research happening there all the time. So check us out. And this is my email address. Feel free to email me if you want to just chat or if you have questions. And also, I'll take any questions now. That's all I've got. Thank you.